Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Experimental Biotechnology, so far we have discussed the different types of techniques and the aspects where you can be able to utilize these techniques and how you can be able to design the different types of experiments to resolve the different types of scientific questions. So, now it is the time to review whatever we have discussed so far in this course and try to refresh our memories, because this is very important that you should recall what you have discussed or whatever we have discussed in this course, so that in case you have any queries or whatever you have not been asked during the previous modules, you can be able to ask me those questions, or in case something which you could not be able to understand while we were going to review the content, you can be able to again get the opportunity to understand that. So, in this particular lecture we are going to summarize what we have discussed so far, and in this process we will try to understand how the different techniques can be exploited and can be used even to answer the single questions or the similar kind of scientific questions. So, we started the course with a good lab practices. So, where we have discussed about the what are the things are allowed and what are the things are not allowed, what are the different types of precautions you have to take while you are going to be enter into the lab. In that particular type of modules, we have discussed about how to how to prepare the different types of buffers or how to even operate the different types of instruments. We have also discussed different types of precautions. So, what we have discussed is that the good lab practices are actually been very important for you to understand that it is not important that you are going to perform the experiment, it is also important that you should perform the experiment as per the prescribed guidelines. So, that is why the people have come up with the idea of the good lab practices, and the major advantage of the if you follow the good lab practices is that whatever you are going to report or whatever you are going to make the discovery through the different types of experiments can actually be able to advance the ongoing literature, and as well as it is actually going to help the others to utilize that particular information, because when you are going to follow the good lab practices, you are actually going to document each and every observations, you are actually going to use the a complete list of material and methods and experimental procedures in your notebook. So, this aspect also we have discussed in detail in this particular type of module, where we said that it is important that you should also maintain a very neat and clean notebook, where you should start it with the objectives. Following the objectives, you have to list out all the reagents and experiments and all the details of the experimental protocols, and then subsequent to that you are actually going to write each and every step, what are the modifications you have done and how long you have incubated the samples and all those kind of technical informations. Followed by that, you are also going to write the observation what you have gone, and then ultimately you are going to get the results. These results could be of the raw result what you are going to directly get from the machine, or you can have the calculated results like the interpreted results. So, in that particular context, we have also discussed that suppose you got the protein estimations, and suppose you are doing a protein estimations. So, one is the raw data like any particular type of absorbance what you are going to get from the spectrophotometer. The other is the background signal what you are going to get from the spectrophotometer. So, if you subtract the background values from your experimental value, then you are actually going to get the calculated value, and these calculated values are the derived data what you got from your raw data. So, it is important that you should note down the raw data as well as the calculated data or the experimental data, so that you can be able to cross verify, or if the people who want to repeat your experiment, the people who want to reproduce the data, they should be able to go through with your notebook, they should be able to understand each and every aspects. So, at the end of this module, we have summarized that you have to follow the good lab practices, which means you have to follow the particular type of disciplines. You have to you should not eat or drink into the labs, because the lab is full of bacteria, viruses and all those kind of infectious organisms. So, that is why you should not consume the food into the labs, as well as you cannot just do as per your wish. So, you have to always wear the safety of instruments, like you have to use the aprons, you have to use the safety goggles, you have to use the other kind of the lead shields and all that kinds of thing. And then if you are handling the infectious organisms, then you have to appropriately use the different types of the biosafety cabinets, like either you use the laminar hoods or do the biosafety cabinets. And that also we have discussed in detail, that what is the different types of biosafety levels are possible and how you should be able to handle those biosafety levels. So, this is what we have discussed, the precautions, what you should do and what should not do, and what is recommended and what is not recommended. And at the end, we have also discussed about how you can be able to design the data register, so that you can be able to document each and every detail of the experiment what you have performed, and so that it should help the other people to follow the content. After that, we directly jumped onto the different types of techniques what you should use to advance the experimental work. So, what we have discussed about the experimental like electrophoresis, we discussed about the chromatography, we discussed about the immunological techniques, we discussed about the cell biology techniques, and at the end we have also discussed about the molecular biology techniques. So, within the experiment electrophoresis techniques, we discussed about the basics of the electrophoresis, how the electrophoresis actually separates the molecules, and how the electrophoretic mobility is directly proportional to the charge and the inversely proportional to the mass, and what are the different factors which actually can regulate the resolution or the separation of the molecule during the electrophoresis. And then following that, we also discuss about the different variants of the electrophoresis, like we discuss about the vertical electrophoresis, the horizontal gel electrophoresis. Within the vertical gel electrophoresis, we discuss about the SDS page or the denaturating gel electrophoresis, or the native gel electrophoresis, as well as the urea page. Whereas in the horizontal gel electrophoresis, we discuss about the agarose gel electrophoresis or the high resolution horizontal gel electrophoresis. While we were discussing about all these techniques, at the end of the electrophoresis, we have also discussed about how the electrophoresis technique can be utilized to answer some of the scientific questions, and all those questions we have taken in such a way so that it should actually not going to help you to understand the potentials of the vertical gel electrophoresis or to the horizontal gel electrophoresis. It also going to help you to design your own experiment, because once you know that this particular technique can be used in all these different types of ways, then you can actually make a combination of those techniques, and you can be able to answer some of the unresolved questions. So, at the end of the electrophoresis, we also discuss about some of the staining techniques, like we discuss about the Comasi brilliant staining techniques, or we discuss about the silver staining, and we also discuss, once you got the images, how you can be able to utilize different types of softwares to analyze the image data, how you can be able to determine the molecular weight of the unknown proteins, or how you can be able to determine the concentration of a unknown protein if you have. So, if you can be able to utilize the softwares, you can be able to get these informations. So, we concluded the electrophoresis module, and then we directly jumped on to the chromatography. Within the chromatography, we discuss about different types of chromatography techniques. So, initially we started with the basic principle of the separations, where we have taken very simple examples of the different types of molecules, how they are actually varying in their physical and the chemical as well as the biological properties, and how you can be able to utilize these physical, chemical or the biological properties to separate them, because using these properties you can be able to distribute distribute the molecules into the different phases. And during that discussion we discuss about how even the simple technique like the distillation can be used to separate the two molecules like the benzene and the alanine, and how the benzene is actually distributing themselves within the liquid phase as well as the vapor phase, compared to that the aniline is distributing themselves from the liquid phase to vapor phase. And this distribution of the benzene and aniline within the liquid versus the vapor phase can be modulated in such a way that the benzene is going to be collected separately, whereas the aniline is going to be separated separately. So, that is just a simple example of showing you the potential of distributing the molecule between the two different phases, that can be even further amplified or further enhanced when you use the chromatography techniques, because in the chromatography technique you are going to use the different types of beads, and all these beads, for example, in a particular chromatography column, if you have the 100 layers of the beads, all these 100 layers of beads are actually going to give you the 100 different types of distribution planes. And because of that, the in compared to that, when we were talking about the distillation, you were only having the two phases, the liquid as well as the vapor phase, whereas in the case of chromatography, if you have the chromatography column with the 100 distribution planes, then you are actually going to distinguish the molecule utilizing these 100 distribution planes, and because of that the molecules are going to be get separated more efficiently into a chromatography column. Following that, we also discussed in detail about the chromatography systems, whether it is a low pressure systems or middle pressure system or the high pressure systems. And then during that process I have taken you to my lab also, where some of my students have also given you a demo about how to operate the purification systems and how you can be able to utilize the purification system. So, what we have given you a demo of the Acta Pure M, but and as I said in the lecture itself, that it does not matter whether you have this particular instrument in your lab or not, because more or less the procedure remains the same, except that you might have to use the different buttons and different things. So, that information you can easily get from the manual of that particular chromatography system, and then with a short span of time you could be able to train yourself to this particular instrument what you have in your lab, and that is how you can be able to utilize that instrument very efficiently. Following that, we have also discussed about the different types of chromatography techniques. So, what we have discussed, we discussed about the ion exchange chromatography, we discussed about the hydrophobic interaction chromatography, we discussed about the gel filtration chromatography, and we also discussed about the affinity chromatography. And within this chromatography techniques, we have also discussed about what is the basic principle of the ion exchange chromatography, what is the basic principle of the hydrophobic interaction chromatography or gel filtration chromatography or the affinity chromatography, how these individual chromatography techniques are actually distributing the molecule or separating the molecules, and how what are the different parameters as well as the factors what is governing their potential to separate the molecules into the different distribution planes. And because of that, we have in depth taken how the ion exchange chromatography, hydrophobic interaction chromatography, gel filtration chromatography as well as the affinity chromatography can be used to design the different types of experiments to answer some of the questions. And while we were discussing about the affinity chromatography, we have also discussed how you can be able to generate the affinity column, how you can be able to utilize that to purify the antibodies, or whether you can use the antibodies to purify the antigen, how we can be able to utilize the metal affinity columns, how we can be able to utilize the pseudo affinity columns and all that. So, with that we have taken up the complete the detail of the chromatography procedures, how you can be able to use the chromatography to design the new experiments, and how you can be able to use that to answer some of the questions. Ultimately, and after that we jumped on to the immunological techniques. So, in the immunological technique, we started with the immunological technique with the first the basic understanding what is the immunology, how the different candidates are playing together and how they are actually giving you the immune responses. And then subsequently we also discuss about the antigens, how you can process the antigens so that you can be able to use that to immunize the animals, and how that can actually give you the antibodies. We discuss about the production of the monoclonal antibody as well as the polyclonal antibody, and in that particular context we have also shown you a very detailed demo how you can be able to immunize a rabbit, and how you can be able to, what are the different steps are required to prepare the emulsions, and how you can be able to check the quality of that emulsion, and how you can inject the emulsion into the rabbit, and then how you can be able to collect the blood, and how you can be able to separate the plasma. And ultimately we have also shown you how to purify the antibodies from that particular plasma. And apart from that, we also talked about the monoclonal antibodies. We have also discussed about how you can be able to separate the B cell and myeloma cells, and all, how you can be able to generate the how you can be able to do the fusion reactions, how you can be able to generate the hybridomas, how you can be able to screen the hybridomas and all that. And after that we have taken some of few of the classical techniques like the agglutination reactions, precipitation reactions, or the in depth we have taken the ELISA also. And while we were discussing about the ELISA, we discussed about the indirect ELISA, direct ELISA, sandwich ELISA, and as well as we have also discussed about the cytokine arrays. So, in the cytokine, while we were discussing about the cytokine array, we have taken you to my lab, how you can be able to perform the cytokine array to measure the different types of cytokines in a single reactions. So, in that case we gave you a complete detail of how you can be able to measure all the 50-60 cytokines what is present into a cell supernatant simultaneously with the help of the cytokine array. Apart from that, we have also tried our level best to show you some of the demos that is been related to the immunological techniques, like we have shown you how to perform the ELISA, how you can be able to measure the antibody level, and so on. After that, we moved on and gave you a full discussion about the cell biology techniques. So, we started with a very basic, that the cell biology techniques started with the cell culturing. So, you can actually have the different types of media. So, we have given you the information about the media, how you can be able to prepare the cell culture media, what are the precautions you should take while you are preparing the cell culture media, how you can be able to autoclave, or how you can be able to do the sterilization of the media, whether it is a microbiology media or the cell culture media. And then we moved on to some of the techniques, like we have shown you the microscopic techniques, like the simple bright field microscopy or the fluorescence microscopy, and as well as we also shown you the scanning electron microscopy or the transmission electron microscopy. Not only we discuss about the microscopy, we also discuss in detail about how to process the samples, so that it should actually give you a very detailed insight whether how to process the sample when you are actually going to design the experiments. So, these are not only the theoretical aspect what we discuss, we also discuss about the experimental aspects, like how to perform the protocols, what are different precautions you should take, and how you can be able to modulate that some of the factors, so that it should actually going to give you the troubleshooting, or some of the factors how you can be able to govern, so that it's actually going to make you more better images and all that. And at the end of the particular cell biology techniques, we have also discussed about the different types of the experiments what you can actually explore. You can, where we were talking about the microscopic techniques or cell biology techniques. Some of the places we have also discussed about the flow cytometry. So, in detail we have discussed about the flow cytometry, how you can be able to utilize the flow cytometry, what are the basic principle of the flow cytometry, how you can be able to utilize that to answer some of the questions, what are the potential of a flow cytometer and all that. So after that we directly jumped on to the molecular biology techniques. So within the molecular biology techniques we started with the very basic, that the with the polymerase chain reactions, and we discuss about the PCRs, what are the how the PCR is being developed, how the technique is being developed. So idea of discussing the development of a particular technique from a very crude status to a very refined level is that to actually give you an idea how the people are actually developing a technique when they are going through a different stages. Like for example, when the people were initially starting the PCR, they are actually using the non-thermostable enzyme, so that because of that they were using the thermal labile enzyme. So because of that, because there was no discovery of the tag DNA polymerase, so because there was no thermal stable enzyme, they were using the thermal labile enzyme. They were supposed to add the enzyme after every reaction. So because of that, it was actually causing a lot of hindrance. And apart from that, the people were also having the very crude kind of thermal cyclers. And so in the crude type of thermal cycler, they were supposed to they were supposed to handle, or they were supposed to transfer the tubes from one compartment to another compartment, and that's how the PCR actually started. But when the people have developed the Peltier based systems, the ramping speed as well as all the other kind of temperature changes were very rapid, and because of that the PCR was becoming a very popular and useful technique. And at the end of that, we have also discussed about how the PCR can be used for answering some of the experimental questions, how the PCR can be used to even detect the different types of the infectious organism or the contamination in a particular sample. So we have taken an example from the agricultural biology, we have taken an example from the plant sciences, we have taken an example from the infectious diseases like HIV, we have taken an example from the hepatitis, we have taken an example from even some of the other viral diseases, and how you can be able to detect the viral diseases, and how you can be able to even detect the infectious organism what is being present into the fruits and vegetable as well, and how the PCR is a very potential technique and robust technique even to answer some of the genetics related questions like who will be the potential criminal and all those kind of thing. And after that, within the molecular biology techniques, we have also discussed about some of the basic blotting techniques, like we have discussed about the southern blotting, we have discussed about the northern blotting, and at the end we have also discussed about the sequencing technique. So we discuss about the DNA sequencing as well as the protein sequencing. So this is what we have discussed so far as a whole, and when we were discussing about the individual modules, we were not considering the other experiments. Like when we were talking about the electrophoresis, we were not discussing about the chromatography. While we were discussing about the chromatography, we were not discussing about the cell biology, immunology or the molecular biology. But in totality, when you are designing an experiment, you actually require the summation of all the techniques what you can do, because when you are actually designing an experiment, you actually require the help from the electrophoresis, you require the help from the chromatography, sometimes you require the help from the immunology, and sometimes you might have to require even the assistance as well as the feedback from the molecular biology. So all these are actually separate techniques, but they are integrative in nature, because these techniques has to be come together, then only you can be able to perform a experiment, you can be able to design the experiment with a more fine controls, because you cannot design an experiment simply with the electrophoresis or simply with the chromatography. That is one aspect. The other aspect is that in some cases when you are actually trying to answer the questions, but you do not have the electrophoresis. So what you can do, you cannot just answer the questions, no, that is not the case. When you have a question, you have to actually answer that particular scientific question by designing an experiment. But since because you do not have the electrophoresis system in your lab, does not mean that you cannot be able to design an experiment. That you can do simply by the chromatography or with the help of the immunology. So that is what we are going to discuss now, where we are going to take the similar problems from what we have discussed so far, but we are going to tell you that the same problem can be even handled with the multiple techniques, so that you can be able to utilize these multiple techniques and you can be able to get the answer. So, irrespective of whether you have the electrophoresis in your lab or whether the chromatography in your lab or not, you can still be able to design the experiments. So, let us see, this is the old technique where we old research problems, where the research problem is that the scientists have isolated a old rock sample with a DNA sample from the dinosaur. They have isolated the DNA and did the PCR amplification with the random primers. Now they want to determine the size of the amplified DNA. So, if you remember, while we were discussing about the electrophoresis, we have taken this problem where what is the problem? Problem is that the scientists have discovered a rock sample where they could found the dinosaur fossil, and when they extracted this dinosaur fossil, they could be able to get some amount of DNA as well. So, what they have done? They have done the PCR, and then they got the amplified DNA, and then this amplified DNA they are trying to identify the size of this DNA. Now, you understand that this is actually one aspect of this problem, that you are actually simply going to run a molecular marker, and it is actually going to give you a calibration curve between the size of the DNA versus the distance what it covered, and then you can be able to utilize the amplified DNA. You can actually calculate the RF values, and it is actually going to give you the size of that particular DNA. But what I am trying to say is that in this particular problem, you are simply using the one of the molecular biology technique, which is called as the PCR. So, you are using a molecular biology technique, then only you are getting this amplified DNA, and then only you can be able to utilize the electrophoresis to answer the questions. Apart from that, you can also incorporate many other techniques, so that you can be able to understand more about this particular DNA. For example, you got this amplified DNA. First thing, you have actually added the a tool from the molecular biology. The second is, once you got this amplified DNA, then you can be able to do a sequencing with the help of either the Sanger's method or the Maxim-Gilbert method. So, you can actually get the sequence. Once you got the sequence, then you can be able to clone this DNA. When you got the sequence, you can be able to do some different kinds of reactions. So, once you done the sequencing, what will happen? You are actually going to get the sequence. Now, once you got the sequence, you can actually use some of the bioinformatics techniques, like you can use the bioinformatics techniques, and that is how you can actually take this DNA sequence, blast it into the genome, and that is actually going to tell you whether this sequence is actually how much it is close to some of the organisms. So, it is actually going to give you a closeness to its relatives. So, that is how you can be able to identify what kind of dinosaur is this, because that information is still missing, because you got the dinosaur's DNA, and but that DNA you do not know what kind of dinosaur, whether it is a Archaeopteryx or whether it is a dinosaur which was closer to bird, whether it is a dinosaur which is closer to lizards, or whether it is a dinosaur to So, those kind of information you will get when you are actually going to do a bioinformatics analysis to this DNA. Apart from that, once you got the sequence, you can easily use and clone this sequence into expression vector. So, you can use some of the molecular biology techniques. You can do a PCR with the primers, the site specific primers, and then you just clone it, and that actually is going to give you the protein. Once you got the protein, then what you can do is you can use the immunological techniques to generate the antibodies, and once you got the antibodies, you are now going to have the another kind of analytical tools which you actually can use. Apart from that, once you got the protein, this protein can be asked for many other kind of experiments, like you can easily ask whether this is an enzyme, whether what could be the activity of this particular protein, whether it is a structural protein or whether it is a functional protein like the enzymes, and so on. So, that is actually going to tell you the potential of the different types of techniques what you can actually integrate in your experiment, and that's how it is actually going to give you the bigger picture of what you have actually. So earlier we were just using the electrophoresis technique, we were just simply doing the RF versus molecular weight analysis, and you are just trying to identify the size of that particular DNA. But now you have the complete information about that particular dinosaur, what you actually having the DNA from. So that is actually going to give you a lot of information and lot of insight into the what kind of dinosaur. And is apart from that, since you have the rock sample also, you can easily calculate the age of that particular rock, and it is actually going to tell you the at what era of in which this particular dinosaur was existing. So that is actually the way you should actually plan your experiment. You integrate all other techniques, and that's how you can going to get a global picture and the full picture about the particular type of problems. Now we have another problem, that is the scientists have discovered a transcription factor and a skin cell bind to the set of gene X, which is responsible for changing its color due to the exposure of light, exposure to the sunlight. Now they would like to identify the transcription factor from the skin cells. So this I think we have discussed when we were discussing about the electrophoresis. And what we said is that you have actually a promoter region. So if you talk about a gene, the gene has two regions. One is the regulatory region, which is called as the promoter. The other one is as the coding region, which is actually been coding for a particular type of protein. So if it is a functional protein, if it could be structural protein, or sometimes the gene does not code for any other protein as well. So this transcription factor is going to bind this regulatory region, and that's how it is actually changing the activity of the production of this particular enzyme. So what you are supposed to do is you are actually going to do the interaction analysis of the DNA region with the protein, and that you can do with simple assay which is called as the gel shift assay. So we discussed that in detail about the gel shift assay, what is the basic principle of the gel shift assay, how the DNA and protein when they are present in a ratio of 1:1, how that is actually going to give you resultant molecular weight which is 2, and how that resultant molecular weight is actually going to reduce its electrophoretic migration onto the gel. So you can imagine that if the DNA is of X molecular weight, it's a Y molecular weight. So if the DNA will run for the X distance, the protein will run for the Y distance, and the DNA protein complex is going to run for the Z distance, then in that case the X is going to be the bigger or the smaller to the Z. So in this case the X is going to be bigger to the Z, because Z is going to be of high molecular weight, because Z is of 2 unit, whereas the DNA is going to be of 1 unit. So the Z is going to be less electrophoretic mobility compared to the DNA alone. And this is what we are doing. We were running the DNA alone, or we are incubating the DNA with a different amount of protein, and what you will see is the DNA alone is going to have the higher electrophoretic mobility, and when you are adding the protein, the electrophoretic mobility is reducing, and that's how you are actually going to have the shift in the electrophoretic mobility. The results what you see is something like this, where you have the DNA alone, and then if you are adding the protein, it is actually the shift of the DNA from the so this is actually going to show you the more electrophoretic mobility, and this is actually going to give you the less electrophoretic mobility. And if you require, you can actually do some of the verification experiments where you can add the unlabeled probe, and that's how it is actually going to compete for the labeled probe, and that's how you are actually going to see the labeled probe coming out from the reactions. Now if you want to do the same experiment, you can also use the ion exchange chromatography, and that also we discussed. So in ion exchange chromatography, what you are going to do is you just take this promoter region, or you take the promoter of this particular gene, and then first you use couple it to the anion matrix, and that anion matrix is going to bind the DNA, and then what you are going to do is you are going to put the protein X in such a way that the protein X itself is going to have the 0 charge. So which means you have to add the protein X onto its pi value, which means if you add the protein X to its pi value, the protein X is going to have the net 0 charge, because that is very important, that the protein X should not bind to the column directly. So in that case, what you are going to do, you are first going to bind the DNA onto the column, and then you are actually going to flow the protein X at a place where the protein X should not have its own charge. And then what will happen, the protein X will go and bind to the DNA, not to the column, and ultimately you are going to elute. If you elute and if you analyze the profile, what you can see is that the protein is going to follow a pattern, the DNA is going to follow the pattern onto the horizontal chain, and if these two patterns are matching with each other, then you are going to say that these two DNA proteins are matching with each other, and actually the protein is binding to the DNA, because the protein itself does not have any affinity for the matrix. So the protein can only bind to the matrix if it is interacting with the DNA. As a verification experiment, you can still do some of the verification experiment, like you can add some of the DNA degrading enzymes, or you can actually change the pHs and all that. So this is what you have to do, is you have to do like a gene promoter, you have to clone, then you have to do the ion exchange chromatography, you have to do the agarose gel electrophoresis, and you have to do the SDS page. So what you can see is that we are using the electrophoresis approaches. We discussed that how you can use the electrophoretic approaches to answer the questions, how you can use the chromatography techniques, and how the same approach can be used even to answer the same questions. Now, what the way it is going to be different? In the electrophoretic technique, you are only going to get the semi quantitative values, like you are going to see whether the protein A is binding to DNA or not, whether that is causing a shift into the band or not, but you could not be able to get a quantitative information. So it is a semi quantitative or the qualitative information what you are going to get from the electrophoresis, whereas from the ion exchange chromatography, what you can also do is you can also get the information whether the DNA and protein are interacting with each other or not. On the other hand, you can also get some of the quantitative information, like you can get the distribution, the binding constants, you can be able to do all those kind of analysis as well. And as you can see, even when we are doing the ion exchange chromatography, you still require the other techniques like SDS page as well as the agarose gel electrophoresis to complement your experiments. So that's why, as I said, not a single technique is on its own, it requires the assistance from the other techniques to complete the experiments. Now we have the third experiment, which is actually Mycobacterium tuberculosis was treated with the drug, and it causes the generation of the oxidative stress inside the bacterial cell. Now the ligand responsible for this effect was isolated, and now the PhD students wants to identify the adapter protein from the Mycobacterium tuberculosis to understand the signaling events and the associated molecular components. Now what is the problem? Problem is that you treated a Mycobacterium tuberculosis, the infectious organism which is responsible for TB, and once you done that, it is actually generating a ligand, and that ligand is going out and then it is binding to a receptor. Once it is binding to a receptor, this receptor is bringing the adapter proteins, and then because of these adapter proteins, it is actually driving a downstream signaling. So what the student wants is he wants to identify these adapter proteins, and if you remember, we have used some of the techniques where you can be able to answer these questions. But in this technique we have discussed, we said that you can just simply make a column where you can actually put the ligands onto the matrix, and then these use as a scaffolding protein, and then if you incubate with the cell lysate, it is actually going to bind the receptor as well as the adapter protein, and then you can actually isolate these components and you can be able to answer the question, so what are the adapter proteins are present, and that is actually going to give you the complete information about the adapter proteins. these are the things what you require. You require recombinant DNA technology, you require ligand productions, you require the coupling to the matrix, then you require the affinity chromatography, and then you require the SDS page to answer the questions. Now let's see a different angle through which you can be able to even answer the similar questions utilizing the cell biology techniques. So in the cell biology technique, what we can do is we can easily look for two things. One, when the ligand is binding, you can actually first characterize that, and also once the adapter proteins are known, like if you are doing that experiment, it is actually going to tell you the information about the adapter proteins. But there are false positive what is actually going to come, because when you are processing the samples and you are processing the whole cell lysate, some of the protein which may not be adapter protein are actually also going to non-specifically bind to your beads. So how to get the information about this? So what you can do is you can simply take the adapter proteins and you can actually do a cell biology techniques, like you can actually first utilize the immunological techniques, you generate the antibodies. If the antibodies are already available from the commercial vendor, then it is fine, otherwise you can generate the antibodies and process the things like that. So imagine that you know the adapter proteins. Now what you can do is you can simply use the adapter proteins and generate the antibodies. Now these antibodies you can use to localize the adapter proteins, and you can also use the antibodies to localize the your receptor. Now once you do that, and they are actually going to be a adapter protein of that particular receptor, they are actually going to give you the co-localization, which means the signal is going to come out from the same point. That is one thing. The second is, once you have the adapter proteins and once you have the receptors, you can also plan an experiment, and suppose you have the antibodies for the adapter protein as well as the antibodies for the receptor, then you can also design an experiment which is called as the fluorescence resonance energy transfer. So in the FRET experiment, it is actually going to tell you the interaction between the different types of proteins what is present within the cell, and that information is going to be more and more authentic and more accurate compared to that when you break open the cells and trying to purify the protein or trying to purify the factors with the help of some of the affinity chromatography related techniques, because that is actually going to give you the opportunity even for the non-specifically bound protein to interact. The third is you can also use some of the proteomics based approach as well to answer the same kind of questions, because what will happen is that what you have two conditions. One, you have the minus drug conditions, you have the plus drug conditions. So what you can do is you can simply take the two cells, one it is control cells, the second is the drug treated cells, and then what you do is you just break open the cell lysate and you prepare the you can just simply take out the membranes from these two bacteria, and then I ask for what are the differentially different proteins are present. So for example, in the control cells, suppose you isolated the membrane and you got the 40 spots, okay, and in this one suppose you got the 47 spots. So what you can do is you can simply do a image analysis and see what are the separate 7 spots are coming up, and whether those 7 spots are could be a potential adapter proteins. So then what you can do is you can just exclusively identify those 7 proteins or 7 spots, trying to identify the proteins with the help of the proteomics approaches, and you extract and get the information about those 7 proteins, and you can be able to utilize that information to cross verify whether those additional 7 proteins could be a adapter protein for this particular receptor or not. So this is the way we should actually integrate multiple approaches, multiple techniques, so that you can be able to answer different questions and you can be able to understand the whole process in detail. Then we also discuss about this problem where we said that the protease PFI 1625C was cloned from the Plasmodium falciparum and all that, and you want to identify the substrate sequence, and we said that you can actually use a peptide coupled to the beads, and then if the protease will digest, it is actually going to generate the different peptide sequences, and then you can actually do these peptide sequence, you can do a multi-mass, and that actually is going to tell you what is the leftover onto these beads, and because of that analysis you can be able to deduce the peptide sequence. So in this one you are going to do a protein production, peptide synthesis, coupling to the matrix, protease assay, affinity chromatography, and then ultimately you are going to do the LCMS. And then what you are going to get, you are going to get the peptide or the peptide sequence what this protease is cutting. But at the end it is not going to tell you the potential substrate what is present in the cell. Now, if you want to do the same experiment and you want to do with the help of the electrophoretic approaches, what you can do also is, suppose, because in this one it is only going to tell you the peptide sequence, it is not going to tell you the potential substrates. Okay. So, what you can do is also, or what alternatively you can do also is, suppose I take the BSA, okay, and I will incubate the BSA with PFI 1625C. Which is a protease present in the Plasmodium falciparum, and then what I will do is I am going to have two reactions, one place where the protease is absent, the other place where the protease is present. And now what I am doing to do is I am going to do the incubation with the BSA in the presence and absence. So, you are going to have two reactions, one the place where you have the BSA, the other place where you are going to have the BSA plus PFI 1625C. Now, what I will do is I will allow these two reactions to run for different amount of time. For example, every 3 hours or 4 hours I will start taking out the reactions. So, what will happen is it is actually going to start giving you the peptides, because irrespective of that you are not adding a protease into a BSA structure or sequence, since you keep the protein into the water, it is actually going to go through with the autocatalysis or autolysis. So, because of that some protein is going to be degraded in due course of time. So, you are going to get the peptides from here, also you are going to get the peptides. Now, what you are supposed to do, you can take these two sequences or these two samples and then you separate them into a using the high resolution page, and what will happen? When you do that, you are going to get the peptide sequences present in both the structures, and eventually definitely when you are having the no protease versus the presence of protease, the number of bands what you are going to see is more compared to this one. So, you are going to, if you do a image analysis and if you do a subtractive analysis, you are going to see that some of the bands are actually common between them, whereas the sum of the bands are actually reappearing and the new bands. Now, once you see that these bands are actually reappearing and the new bands, then what you can do is you can simply take out these bands from the gel with the help of the isoelectric elutions, or you can be able to use some other technique to extract these protein bands, and then you can simply go with the proteomics approaches and identify these peptide sequence. Now, these peptide sequence, so if you suppose this is a protein, right. So, if it has been chewed up for multiple places by the PFI 1625C, this is the peptide what you are going to see into the sample. So, once you have the ends of this particular peptide, so if you use the protein sequencing method or the proteomics math approaches, you are actually going to get the sequence of this peptide. So, once you get the peptide sequence, and you are not going to get one peptide sequence, you are going to get the multiple peptide sequences, you will be able to deduce from where this particular peptide is coming off, because you already know the sequence of the BSA. So, from there you can say, okay, this is the place I am getting, this is the place I am getting. This means you are going to get not only one peptide, you are going to get the multiple peptides, and that is how if you do all that analysis, it is actually going to tell you the preferences of this particular protease or the site what it actually required to cut the proteins. And that is how you can be able to deduce not only the substrate peptide sequence which it actually prefers to cut the protease by the protease, or it also going to tell you whether the BSA is a standard substrate or whether BSA is going to be a substrate for this protease or not. I am just giving you an example with BSA, but if you require, you can actually use some other proteins also from the host cells or from the parasite cells. So, this is all about the course experimental biotechnology. So far what we have discussed, we discussed about the different types of techniques. We have also summarized you good lab practices, what we should not do and what should do as well. At the end, we have also discussed about some of the different experiments and how the multiple approaches can be used to answer these questions. And so, with this I would like to conclude my lecture here, and I hope you will actually understand the content, and I hope that you will be able to follow these experiments, and I hope that this course is going to advance your work. So, with this I would like to conclude my lecture here. Thank you.